and welcome to the Beyond Biotech podcast, and this is number 20, How Time Flies. It's also being put together on the road, with a little bit being done in Leipzig and the rest in Berlin. I'm Jim Cornell from Labiotech, and it's been busy. It started with a 2 a.m. alarm clock. Driving the hour to the airport was fun because there was thick fog. And then it was to Amsterdam, where I embarked on the marathon train journey, which really wasn't so bad. It was long and a little bit tiring with lots of changes, but I do have to say that the DB app, which is the German train app, actually does do a pretty decent job of simplifying it. And I didn't have to wait long on this trip to be asked for directions. It happened at the platform in the Netherlands when someone asked me in German which train to get to go to Bielefeld in Germany. And fortunately, it was the same train that I was getting, although I wasn't quite going as far as Bielefeld. I'm really not sure, though, how local I looked with a baseball hat and a US college hoodie on, but there you go. I have to say Leipzig is an interesting city. Lots of history, a lot of modern architecture, a bit of East German legacy, and a whole load of graffiti, and very interesting crossing lights. I stayed in a neighborhood where I had the feeling if I stayed still for more than 15 seconds, I too would have had some stereotypical graffiti initials daubed on me. But if you're a classical music lover, you could do worse than visit Leipzig. It has connections with a lot of the greats. Bach is buried there. There's a Bach museum, a Mendelssohn museum, and another one honouring Grieg, who wrote Peer Gynt there. And there's even an instrument museum. There's a Schumann connection, Mozart too. In fact, I think that every composer seems to have a connection to Leipzig. As I wandered through the streets and the parks, I was expecting inspiration to strike and for a melody to just pop into my head out of nowhere. But sadly, music doesn't always seem to work like that. You can never guarantee when you'll get an idea. I do remember once getting a song in my head, both words and melody, which was unusual, while walking to do some photos when I was editor of a newspaper, and I had to run back to the car singing the song so that I could record it on my little tape deck. Of course, this was long before every phone in the world seemingly had a voice recorder. Anyway, I was impressed with Leipzig. Of course, the actual reason for being there wasn't for the composers, but it was for Bio-Europe at the Leipzig Messe, which really isn't very close to the city centre. Fortunately for walking, the weather was nice, but I did do a lot of interviews there, a lot of walking, and lots of meetings, and probably far too much talking. I talked about baseball, hockey, music, travel, and even squeezed in some biotech as well. And I saw some people that I'd met at NLS days, and other people I'd met before, which was nice. Someone saw my name tag and complimented me on the podcast. I think they said it's great, not it grates. I did have one big mishap when I mistakenly pressed the wrong button when recording an interview, and I did it in slow-mo. I didn't know that until nighttime when I was uploading all of the videos, and it said that the interview was 1 hour and 53 minutes long. So when I hit play, it was a bit like watching someone doing Tai Chi. And you would think that it would be an easy fix, but it took me about 2 hours to figure out how to make it right. And from the event, it was on to the German capital, Berlin, to meet up with my colleagues, who, for all I knew, were either all very tiny or very tall, because you just don't know when you only talk to them every day on a screen, and you never see anything other than their head. It's almost like first impressions for the second time. Anyway, here I am in Berlin recording the podcast, and now I should tell you who's on it. So this week we have François Colery, head of the Talent Now initiative, and Sylvie Ponchaut, managing director, both from BioWin, eTherapeutics CEO Ali Mortazavi, and Keith Fargo, chief scientific officer at the CMT Research Foundation. And we also have a weekly chat with global commercial real estate services company JLL, and this week it's with Robert Coughlin. This would be the time when I politely ask you to head over to Biotech.eu to check out all of the week's news. And as we've all been in Leipzig and or Berlin this week, it's probably best to skip it this time. Although what I will say is that there are at least a couple of video interviews from NLS Days in Sweden, and those are with the winner and the runner-up of the NLS Invest pitching contest. So definitely worth heading over to Labiotech.eu for those. So let's get to this week's interviews. eTherapeutics is a company integrating computational power and biological data to discover life-transforming RNAi medicines, and it recently announced a fundraise of £13.5 million. To tell us about the company, what it does, and what the money means is the company's CEO, Ali Mortazavi. So I guess the first question then is if you could tell me a bit of background on eTherapeutics.
Yes, Jim. So eTherapeutics, maybe a little rewind back to kind of where the sort of genesis of eTherapeutics is. I've been in the field of RNA therapeutics, specifically RNAi, for about 11 years, and was previously CEO of a listed company called Silence Therapeutics. Around, I guess, 2017, 2018, there was a sort of major technical breakthrough in the field of RNAi, where at least delivery of RNA molecules to the liver has been largely, let's just say, solved, which really completely changed the industry, which had maybe for 20 years or even longer been struggling to make any impact at all. So we developed that system also at Silence. And I guess the next sort of stage of that is any new drug platform is extremely rare to come by a drug platform. I mean, something that is, if you like, modular, is capable of, has been validated by approved drugs. There are about 4 or 5 approved drugs in the field of RNAi in the liver now, so a good body of safety and efficacy. I think it's fair to say that by joining therapeutics in 2020, by that stage, we're talking about a new validated drug platform of which I've had a lot of previous experience in. Kind of leaving Silence Therapeutics, we spent so long trying to develop this piece of chemistry, if you like, drug platform with the relevant IP, but kind of walked into the next big problem in drug development, which is understanding biology and in effect, good drug targets in the liver. And that's where I kind of bumped into a company called eTherapeutics, who'd been very active in the field of computational approaches to biology. So I kind of saw an opportunity to marry up what I think is a huge opportunity in terms of a drug platform in chemistry, and to be able to have a really differentiated strategy in using computation to find biological drug targets and really understand biology better. So it's kind of 2 pieces of jigsaw kind of coming together. And I guess that's the sort of starting point for the strategy going forward at eTherapeutics, starting in, let's say, March 2020, actually a week before lockdown.
Can you give me a bit of a detail on what kind of things you work on?
So the RNA chemistry platform is, if you like, we had to develop our own IP here at eTherapeutics. That's been done. We've got multiple patent filings. We know that we can deliver an RNA to one specific cell type called hepatocytes within the liver, which is the vast majority of liver cell types. I guess the next layer of that is the liver and hepatocytes is such an active organ in kind of human disease. So, for example, inhibiting target genes in the liver aren't just relevant for liver diseases, they're relevant in blood disorders, in cardiovascular diseases, and that's a function of the fact that the liver is, if you want, the central sort of trafficking system of the kind of human body. So we narrowed, if you like, to one cell type, but in that narrowing, the target market, if you like, and opportunity space is still very, very large. So the next aspect was to kind of reformulate the old eTherapeutics computational biology platform to be highly focused on what we call hepatocyte biology, because that's obviously, we know our drug can go there. We know that we can, in effect, inhibit any target gene in hepatocytes. And that's been a process which is obviously ongoing. That's something kind of what we call the hepatocyte atlas. And really the job of the Hepatocyte Atlas is, as I said, to try and find differentiated novel drug targets. So obviously computation always relies on very large data sets. We have one of the probably largest hepatocyte-specific data sets out there. And the next kind of level of that is algorithms and techniques to be able to model biology in a computer, in effect. So kind of what we're trying to do is, as much as possible, bypass that sort of very difficult time and costs of traditional drug development by looking at biology almost as a sort of data science problem, if you like. Because of the function of hepatocyte biology in disease, we are active currently in blood disorders, cardiovascular disease. We're looking at NASH space, and we've got active early stage programs going through our process as we currently speak. So I guess the next layer of that would be, how many target genes and potential diseases are there? What is our sort of, if you like, market opportunity? And as I said, that's extremely large in terms of numbers of genes which are correlated potentially to diseases. And the next layer, which I think is very interesting for this drug platform, not only is, as I said before, a drug platform is hard to get your hands on and know and have the IP. So we've obviously got a lot of in-house experience from people who've been in the industry at Silence, etc. The kind of next aspect of that is to sort of minute we nominate a target and a particular disease from our computational outputs, it takes us about 6 to 9 months to make the drug and get a proof of concept in a biological system such as a mouse in a mouse disease model. Now that is like quite a remarkable timeline. In terms of costs, it's kind of very, very small. Just to give you a comparison, the same process in small molecule drug development, just to make a drug, small molecule drug, could take many, many years. It's very, very hard to actually characterize that drug and can cost many millions. So that's just the drug component. Our drug component for a particular target in biology is in the hundreds of thousands of dollars and takes months rather than years. So it means that we can, in effect, almost apply a totally different sort of business model to kind of drug development, in the sense that everything is just so much faster and more modular. It's a kind of rapid in vivo kind of testing. To take it further, to get to a phase 1, we think we can do that in 18 months from the minute we say kind of go from the outputs from the computation. So again, if you want to compare to, let's say, small molecules, to go from idea to beginning of a phase 1, many millions of dollars, can take 4 or 5 years. The risk is greater. In our RNA platform, like I said, to go from idea to initiation of a phase 1, 18 months, in the very, very low 7 figures. So you get this kind of very nice blend of speed, specificity, because our drug is specific to one gene, which kind of means that it's more translatable from a mouse to a human, and the time that it takes to really get a go, no-go answer, which is, when you put those sort of variables in the mix, is the kind of holy grail of what drug development is. It's kind of fail early, fail cheap, if you like. Reverse it, succeed quickly, if you like, is the other way of looking at it. So, that's, to me, a true definition of a platform drug company, something that you can have these attributes that I've kind of talked about and have a large target market space to aim at.
Do you collaborate with other companies, or is this all just on your own?
For us, remember, like, the timelines are so fast. So I joined eTherapeutics like a week before lockdown, March 2020. We developed our RNA platform, I'd say within 18 months, which is lightning speed and file relevant IP. And we're already prosecuting targets and diseases. One of our sort of, I wouldn't say problems, but one kind of, we haven't been doing this for very long. And, really probably the minute, the first day that I think within eTherapeutics that eTherapeutics could say, okay, we have access to this RNA platform and the relevant IP was probably around October, November of last year. So the lights, if you like, came on then. The answer to your question is we have more to do than time and money, and, even almost unlimited money. There's too much opportunity. Collaborations are a big part of our business model. Collaborations can come in the form of people having access to the platform. The drug platform can come in the form of people that have, let's say, proprietary data in hepatocyte biology, which we may not have, that we may be able to mine for them or kind of set our algorithms loose on and be able to model biology. And or sort of kind of any sort of mix and match of those kind of functions. So no, yeah, it's a kind of hybrid approach. We obviously want to take our most high conviction ideas as far forward as possible, but at the same time, what we're really trying to sort of focus on is repeatability and speed. So if we can sort of get to a process where every few months we're getting data readouts, we can assess the data as it comes out very quickly. That, I think, is our number one goal, is show repeatability, process, and the computational platform generating, tractable good ideas, if you like.
With all of the things that you're working on, where are you currently at with those? I assume they're all in different stages.
So kind of, we're sometimes victims of our own speed, but we are now have 4 targets in 3 indications coming up to animal disease model potential readouts, so it's being staggered like that. We have many more new targets and indications going through the hopper process. So really the kind of ball has got rolling in the last 6 months to a year, in the sense, pretty difficult global conditions, I would say. We didn't quite sort of cater for, we walked into the kind of Omicron process, the work from home debates, the whole capital world markets, and et cetera, et cetera. We are basically in that sort of funnel process. We will be getting hopefully readouts as regularly as possible over the next months, years, and that's kind of what we're trying to do, get clear go, no-goes in clean biological readouts. Some will go to phase 1, some might get paused because they're not good enough, some might be partnered. That's the kind of decision points we're trying to get to.
I guess the most recent news that came out of the company was raising some more funding, which was 13.5 million. What does that allow you to do?
Well, so, our main costs are the prosecution of targets and indications. And so that will allow us to be able to really kind of prosecute our best ideas and multiple versions of them. So really it's a function of doing more and getting, as I said, readouts every quarter, so that we can basically try and sort of computerize, try and get as close to sort of the speeds you might expect computationally. I mean, it's not obviously that fast, but if we can get to from an idea to a readout in 6 to 9 months and be doing that in kind of multiple cycles, that money really kind of powers that. If you think it takes something like, it's slightly different per idea, let's call it $500,000 to $800,000 per target. In simple kind of maths, we have bandwidth now for 10 more of those, if you like. And I think, if we can prove this process out to be a kind of legitimate process for doing things faster, cheaper, and assessing risk better, then I think the access to capital will grow from that.
Looking into the crystal ball, what do you see for the company in the next short term and long term? What are the goals?
Yeah, I mean, kind of in the very short term, I think it's just to get, to really kind of optimize our data sets and algorithms to really kind of get this, let's call it hepatocyte atlas, to really sort of differentiate us from potential competitors. And the next stage is the execution of, from target or idea nomination, to be able to show the repeatability of being able to do it in the timelines and costs that we've seen. And medium term, long term, I mean, ultimately, I kind of joined this industry because drug development is hard. There are a lot of unmet medical needs. And, at the end of the line, there's a very big societal problem in terms of how long it takes to make drugs for patients, how good those drugs are, and, on top of that, the cost of actually making and providing those drugs. So that's, obviously a very big medium, long-term goal. But I think the kind of foundation of it is that beginning process, because then I think we really do look to solve those really kind of sort of structural issues of drug performance, speed to patients, drug pricing. We're taking it bite by bite to basically get there.
Is there anything else that you wanted to talk about that we haven't?
Our chemistry platform, i.e. we can or, with pretty high confidence, we can always deliver the drug to hepatocytes. There are, let's say, X number of genes that are basically we can target and indications. And I think an important sort of phrase, if you like, is we want to make sort of each time we do something not a new problem. So kind of in the world of, let's say, small molecules, every new project is completely different to the next one. Whereas, the kind of core hypothesis here is that every new project is actually very similar to the other process, to the other project. The difference being maybe you replace a gene and an indication. But we're trying to kind of get that kind of almost bringing the disciplines of engineering to drug development, which I think is ultimately going to have the maximum benefit, as I said, in terms of impact of drug performance, real unmet needs being met quickly. And ultimately, our cost of goods is pretty low. So if we are successful, in kind of phase ones and forward, we have a lot of levers, if you like, to really price things correctly is the ultimate kind of end game, which I think is something which is a global problem in any drug modality.
Next, we are Belgium-bound to talk to BioWin, which is the health cluster for the region of Wallonia. Its Talent Now program is aiming to tackle the recruitment challenges for the biotech and medtech industries in Belgium. And to tell us more are François Colery, head of the Talent Now initiative, and Sylvie Ponchaut, the BioWin managing director.
My name is Sylvie Ponchaut. I am the managing director of BioWin since 2015. I am a pharmacist by training and I have a PhD in biochemistry.
And I'm François Colery. So I joined BioWin early this year in January. I have more of a business background and a learning and development background, and I started at BioWin to lead the project Talent Now for the Wallonia region. Yes, I say I'm a master in economics, and I've worked in the industry for almost 20 years now.
To get things started, as if you could give me a bit of background on BioWin.
BioWin is the health competitiveness cluster of Wallonia in Belgium. BioWin is clearly the regional reference for all stakeholders active in the biotech and medtech sector. We currently have 250 members. We have large companies like GSK, Baxter, Kaneka, etc. We also have many startup companies coming from the universities, as well as the innovative SMEs. The cluster strategic areas of expertise are biopharma and industrial production of biomedicines. We include there cell and gene therapy products. Number 2, medtech. And number 3, digital health.
You mentioned that you cover Wallonia. Do you have much contact with and interaction with the other regions, like the Flemish region and the Brussels capital region?
Yes, certainly. Of course, it's a regional cluster, but we have many contacts with Flemish counterpart, simply because they have also their critical mass of expertise in the pharma sector with top-level scientists in research institutes like VIB, for example, and large and small innovative companies. So a lot of contacts with Flanders and contacts with some companies located also in the Brussels region, most of them in the Brussels region, active in the medtech sector as well as in digital health. So many contacts with different regions in Belgium. We also have a strong relationship with other clusters in Europe, in Paris, for example, Medicine, in Lyon Biopole in France. And we are involved in different activities with many biopharma clusters in Europe.
I guess one of the things that you've introduced is a program to tackle recruitment. Before we talk about that program that you've introduced, I wonder if you could tell me what the current issues are surrounding recruitment.
Yes, the issues around recruitment in biopharma are really various. First and foremost, it's the volume of open roles within the industry, which is really high. We are looking on an annual basis for 800 to 1,000 roles for the biopharma industry in Wallonia alone. Most of those roles are within bioproduction and for profiles with high school degree or bachelor's degree. The next roles in demand are, no surprise, within R&D. Then comes data scientists, quality assurance, and regulatory roles. And we don't see those figures decreasing in the short term. Secondly, some roles, even if they're not big in volume, are more and more difficult to find, especially if some work experience is required. It's not uncommon for companies to look for 6 months to a year to find the right candidate. And finally, the profile of the candidate is not always aligned with what the industry is looking for. Hence, the need to offer a continuous education relevant and in line with the latest scientific and technology development.
Is this something that's unique to Belgium? I assume not, but if not, are there any specific issues that need to be addressed or any that are more urgent than others?
Yes, and of course, this is not specific to Belgium and it's observed throughout Europe. As Sylvie mentioned, we exchanged with our counterpart in France, and we have seen recently that they face exactly the same challenge around recruitment and continuous education. I would say the specificity of Belgium is the weight of the sector per capita versus large countries, with the consequence that the cohorts of the graduates coming out of scientific and medical education and who apply for jobs in our industry, that those cohorts are probably not enough to close the gaps. Another issue for Belgium is also the split of responsibility amongst the different levels of governments in Belgium, so federal versus region, Wallonia versus Flanders. And it's an issue and increases the difficulty to coordinate and align efforts to tackle the talent issue.
And so a part of that was you launching the Talent Now program. Could you tell me about that?
Yeah, the Talent Now program is a 3-year program project built around a consortium of the 6 largest biopharma employers in Wallonia. Those are UCB, GSK, Thermo Fisher, Takeda, Janssen, and Universals. The objective of the project is really to develop short to medium-term activities to answer partially, let's be realistic, this talent crisis. The project activities will answer the needs of the consortium, but we will share them with the other small and medium-sized companies in Wallonia in order to give a boost to the entire ecosystem. So for the project, we are developing activities around 3 axes. First, competencies development. So we are looking to simplify the continuous learning offer, to build training to answer the unmet needs, and to build bridges between academia and industry. For example, to prompt expert workers to become trainers in training centers. Secondly, we put an emphasis on the recruitment strategy by bringing together the industry and the regional employment agency to capitalize better on existing matching tools between offer and demand and to explore new approaches. And thirdly, we are working on the sector attractivity, trying to enhance our visibility throughout numerous actions, job days, open days. We communicate more on social media to bring more young people into technology and scientific education so they become the rich pool of candidates for the future for us.
And could you tell me a little bit about the EU Biotech campus?
Yes, the EU Biotech campus is probably one of the most strategic investments of the Belgian Recovery and Resilience Plan. This plan was approved by the European Commission in July 2021. The campus will be located in Gosselies. It's not far from Charleroi in Belgium. And this European Biotech campus will be a unique multi-operator and state-of-the-art training center. It's also a business accelerator. It's an international flagship project. and it aims to provide cutting-edge infrastructure for outstanding talent development with the newest digital and biomanufacturing skills in biotech and biopharma. The development of the facilities has just started, and the campus is expected to be operational in 2025.
So I guess then it will be addressing some of the issues, not just in Belgium, but across Europe as well.
Exactly.
Yes, and it will be an European outreach for students and employees.
Obviously, it's a problem in every industry, but how are you going to be able to attract people to the program?
It's a challenge for the European campus, and I think that in the team, a lot of efforts will be dedicated to communication, promotion, and also to collaboration with other regions and campuses in Europe.
The people that will actually go onto this program, would it be university students leaving university and going into a program like this, or would it be children leaving high school? What kind of age range do you think it will be?
The target audience is really broad. It will be indeed students from university. It would be also the workers or people already working within the industry wanted to develop the competencies in the new technology or in the new scientific fields. It will be also job seekers within the Wallonia region, but also Belgium and from Europe. One of the activity that the campus will also develop is what they call STEM immersion or STEM attractivity. So it will be also targeted at younger children or young teenagers to make sure that they find science and technology attractive and they embark into an education into science, technology, or even mathematics or engineering.
Yeah, I guess there's a lot of different talents and skills needed within biotech and biopharma. It's not just being in a lab and working with test tubes. There's so many different aspects to it, but I guess that's part of the need to communicate what those positions are.
Yes, exactly. We are really looking for people from the workshop in the manufacturing lines up to PhD that works into the labs and discover the new drugs, the new vaccines, the new technology for the future. And that's really the challenge of talent now of Belgium and Europe, is that we need everything and we need them in quantity. So that's really the challenge for the future.
Now we have a conversation with Keith Fargo, Chief Scientific Officer at the Charcot-Marie-Tooth Research Foundation, which recently made its biggest ever investment to Samsara Therapeutics. Keith can tell us more. Before we even talk about the CMT Research Foundation, I wonder if you could brief me on what Charcot-Marie-Tooth actually is.
Yeah, I'd be happy to. Charcot-Marie-Tooth disease has nothing to do with teeth. So that's sort of the first myth, that I'll dispel. Charcot-Marie-Tooth disease is actually an umbrella term that incorporates any inherited peripheral neuropathy that includes both sensory and motor problems, essentially. So if you have a neuropathy that you have inherited from a parent, sometimes it's even actually a de novo mutation, but it has to be a genetic mutation that leads to the neuropathy. It's not just, diabetic neuropathy or chemotherapy-induced neuropathy. It's neuropathy that's driven by a genetic mutation and that affects both sensory system and motor system. And the reason it's called Charcot-Marie-Tooth actually is that it's named after the people who first described it about 135 years ago. There were two French neurologists named Charcot and Marie. And then there was also a British neurologist whose last name was Tooth. And these folks described the disease essentially at the same time. So Charcot and Marie published a paper, and then Tooth published a paper. And so now they're all three given credit in most places in the world, hence Charcot-Marie-Tooth. In terms of the prevalence, it's about 1 in 2,500 people. So that is about 3 million people around the world. And in the United States, it's somewhere between 125,000 people and 200,000 people.
So still pretty significant. Is it easy to diagnose?
Oh man, that's interesting. So it's not easy to diagnose. That's probably mostly because it's technically a rare disease, right? And I think there are different definitions depending on where you go in the world. But in the United States, the definition for a rare disease is fewer than 200,000. So we do fit the definition of rare disease, although which comes with some benefits actually to pharmaceutical companies working in the area. But we're actually the single most common inherited neurological disease. So even though we qualify as a rare disease, it's a pretty common disease among those rare diseases. Now, that being said, most people with some kind of a neuropathy, it's not due to CMT. And so there is kind of this diagnostic journey that people go through where you have to rule out things like diabetes, injuries, etc. And then the way that CMT is typically diagnosed is it's a clinical genetic diagnosis. So you're looking for neuropathy, you're looking for deformations of the feet and then hands actually as time goes by. There are some pretty clear signs having to do with the shape of the feet and the legs in particular that they used to call, and this, I apologize for even using this term, but they used to call stork legs or the inverted champagne bottle sign. And that's because you begin to lose muscle tissue in the feet and lower legs. And as the muscle becomes atrophic, it gets, much, much smaller. And so you get these sort of, they look very distinct, let me put it that way, compared to people who do not have Charcot-Marie-Tooth. But then finally, you're going to want to use nerve conduction testing as well. And you can broadly distinguish two sort of main types of Charcot-Marie-Tooth disease. There's type 1, which causes lowering of nerve conduction velocities because the problem starts with losing the myelin in the peripheral nerve, and it's the myelin that allows you to have rapid nerve conduction. So as you lose that myelin, your nerve conduction velocities slow. That's type 1. Type 2 CMT, the problem really begins in the neurons rather than in the Schwann cells. And so in that case, you typically don't see decreased nerve conduction velocity, but rather you see lower amplitudes of the complex motor action potentials. Now, that's all kind of like neurophysiology gobbledygook, but what it gets down to is slower nerve conduction is type 1. Whereas reduced amplitude or size of the signal, you can think of as type 2. And then finally, for many, but not all types of CMT, we know the genetic mutation that leads to that type of CMT. And so for most people, you can sort of get a definitive diagnosis, if you will, through a genetic test. But that won't pick up all cases because there are some subtypes that we don't know the genetic mutation for yet.
In terms of recognition, I admit I'd never even heard of Charcot-Marie-Tooth. How much of a difficulty is awareness?
Awareness is huge. And one of the things that shocks people when they learn about CMT is that it has essentially the same prevalence as multiple sclerosis. And people find that it's 3 million people across the globe for both diseases. And people find that really shocking because they're like, well, I know 1 or 2 people typically with multiple sclerosis, but most people think that they don't know anyone with Charcot-Marie-Tooth. Chances are they probably do. Actually, before I started working at the CMT Research Foundation, I spent 7 years working at the Alzheimer's Association. And there was another fellow there who worked with me. We overlapped for 4 years. And I never knew he had CMT until I left the association to come work at the Research Foundation. And at that time, he said, oh, well, I have CMT. And it was a big surprise to me. And that's because you can largely hide your Charcot-Marie-Tooth disease, especially early in the disease. And it's a very slowly progressive disease. So, for example, if people aren't seeing, your legs, if you're wearing long pants most of the time and you wear braces, people may not see, sort of gait unsteadiness. They're not going to see the shape of the leg changing. But then over time, as people age and the disease gets worse, because it is unfortunately a progressive disease, typically then you'll start to notice things that make you think, oh, well, something's wrong, but you may not know what it is. And so it's very common for us to, meet people who've never heard of Charcot-Marie-Tooth, even though they may actually know some people with the disease.
So moving on to the foundation, I wonder if you could give me some details on the CMT Research Foundation.
Yeah, I'd be happy to. So the CMT Research Foundation is about 4 years old now, and we have one focus, and that's to deliver treatments and cures for CMT. I mentioned that CMT is a genetically defined disease. Everyone who has CMT, they have it because something is wrong in their genetics. And of course, we're now living in the era of genetic medicine. The first gene therapies were approved 5 years ago now, actually. And so a lot of the tools that we think will lead to straight, not just treatments, but straight cures for Charcot-Marie-Tooth are sort of at our fingertips now. We have to figure out how to bring those to bear against this interesting and difficult nerve disease, because the issue, what makes this difficult isn't that it's genetic. What makes it difficult now is that it's really focused in the peripheral nerves. And that's not an easy tissue to target. If you have a disease that is centered in the liver, it's extraordinarily easy to target the liver. And so we're seeing some gene therapies now actually that are targeting cells in the liver, but targeting the nerves is a much more difficult prospect. And so that's why we were founded, because we think that we're there, but we need sort of a push to get over that finish line with gene therapy.
You mentioned the fact that there's prevalence globally. Is the mandate of the foundation national or global? And in those terms, are you making partnerships with other organizations?
Yeah, so we're absolutely a global organization. We think all good science these days is global in nature. We're living in the era of big science, team science, collaborations are absolutely critical, especially for a rare disease, because even though, like I said, there are probably about 150,000 people in the US with CMT, there are 3 million worldwide. So with those numbers, we can do science that we couldn't do if we were only focused in the United States. So many of the members of our scientific advisory board are from other countries. We've had members from Italy. We currently have a member from Australia. We have a member from the United Kingdom. So just all over the place. And the same thing is true with our funded research projects. So we have funded a company that is based in the United Kingdom. We have funded 2 projects in Italy. We've funded projects in Germany. Most of our projects are funded in the United States, but we have representation all around the world. And we expect that to continue to grow as we continue our work.
And you have a research convention. Can you tell me a little bit about that, what it does and who attends that?
Yeah, I would love to. So we just had our second research convention in September of 2022. So that's also global. Actually, the name of the convention is, the way we name it is we have the year and then the Global CMT Research Convention. So this was the 2022 Global CMT Research Convention. And I haven't looked, we just came out of it, so I haven't looked at all the numbers in terms of attendees, but I can tell you that last year we had attendees from over 30 different countries who attended the convention. So that was great. Our goal with this convention is to have a single research convention that is focused only on Charcot-Marie-Tooth disease and that will attract people from multiple different disciplines. So I joined the CMT Research Foundation in 2020. And one of the things that I noticed right away was that we have people who are doing CMT research who attend the Society for Neuroscience annual meeting. There are people who attend the Peripheral Nerve Society annual meeting and, a few others as well. But in terms of a single convention focused on one disease, that was just not happening. And I can tell you from my experience at the Alzheimer's Association that having that kind of convention where it's disease focused and pulls people from all different clinical, basic science, translational research disciplines, for-profit companies, biotech, pharmaceuticals, getting all of those people together in the same place, focused on a single disease, is really catalytic for the entire disease research area. And so we felt that was important for us to establish in CMT. And so far, so good.
you wonder why more conditions and diseases don't have that same kind of focus, because clearly having something like that would break down barriers, and a lot of people working in silos, it would break those down too.
We think it does. I mean, we had more than 30 biotech and pharmaceutical companies were represented at the convention, which was just terrific. Previous conventions that we go to have that do have CMT as part of what they're focused on. We see a lot of academics there, but not so many biotech and pharmaceutical companies. Part of what we're trying to do is get new people thinking about and researching Charcot-Marie-Tooth disease. So attracting the biotech and pharmaceutical sector was very important to us for this conference. And already we've had collaborations coming out of that, with biotech companies collaborating with academics who are working in CMT. And that's exactly what we're trying to do, is just speed up the pace at which this research happens.
And so could you give me a kind of an overview of the kind of research that's taking place right now and where we're at in terms of treatments?
Yeah, I would be happy to. So most of the treatments that are furthest along right now are actually small molecule therapeutics. And so you've heard me talk a lot about gene therapy, which is a major focus for us, but it's certainly not the only focus. So gene therapy, we think, is close, but it's not going to capture every person right away, especially people who have more rare subtypes of CMT. And so we continue to support research that is based on keeping the axons of the nerve healthy. There's evidence in the literature that even if you lose Schwann cells, which make up the myelin, it's really the secondary loss of axons that causes disability. So we think there's a real possibility for helping people by keeping those axons healthy, and even in some ways keeping that myelin healthy without necessarily targeting the underlying genetic disease. So right now there are 2 phase 3 clinical trials happening in Charcot-Marie-Tooth disease. One is for the major subtype, which is CMT1A, that accounts for about half of all people with CMT. And another one is for a relatively rare subtype that doesn't even have its own designation yet. There's sort of a alphabet soup, CMT1A, 2C, 4J, etc. The type that I was just mentioning doesn't have one of those indications yet, or one of those names yet, because it was only discovered 3 years ago. And that's SORD, S-O-R-D, SORD deficiency. So these are people who have a genetic mutation in their SORD gene that leads to CMT. And there's a small molecule phase 3 clinical trial underway right now looking at treatments for people who have those SORD deficiencies. Behind that, you have some people who are working in cell therapy, and then you have numerous people who are working in genetic medicine. And the genetic medicine is really just taking off for Charcot-Marie-Tooth now. I'm not sure that any of the genetic medicine folks actually are in clinical human trials yet. There may be some that are sort of a hybrid of cell therapy and gene therapy, but we expect the first major gene therapy trial to start in 2023, actually. And so for looking at gene therapy treatments, we're probably, somewhere in the late 2020s, potentially 2029, 2030, before we would get the first approval for a gene therapy. In terms of the small molecule, since those trials are already ongoing, we think we could see an approval there, depending on the outcome of those phase 3 trials. We think we could see an approval there potentially sometime in late 23 or early 24.
It's good, yeah. Certainly some of those are quite close, yeah.
They are. And, it's been a long time coming. But again, that's part of why we founded the Research Foundation, is because we think that it really is time now for Charcot-Marie-Tooth to start to benefit, from some of the trailblazing that's happened in other diseases.
And as far as those treatments are concerned, there's lots of different options and lots of different things happening, as you just mentioned. But are we looking at sort of alleviating symptoms or slowing the disease down or an outright cure?
Well, ultimately, we're going to be looking for outright cures. And that's only going to be the case, of course, in subtypes where we know the genetic problem, right? And we know the mutation that is causing the disease. That gives us something that we can potentially target to cure the disease. In other cases, especially with the small molecules, even if they're disease modifying, we're really looking there at mostly reducing symptoms, potentially slowing, maybe stopping progression. Based on the animal literature, there's some thought that you might be able to reverse some of the CMT symptoms, but that's based on the animal literature, and we don't know what's going to happen in humans yet. So I just want to be a little cautious to anybody who, might be listening to this and has CMT. We really just have to wait for the outcomes of those trials. But the animal literature looks quite good in terms of even being able to reverse some of the symptoms of CMT.
You work with funding of projects. How involved are you in that? Is it just a case of here's the money, off you go, or do you stay involved with the projects?
Great question. It's a little bit of a hybrid between those two things. So, we tend to look at this like we're sort of bringing expertise in the disease, in Charcot-Marie-Tooth, and knowledge, but we're out looking for people who have expertise in different technologies and different assets, gene therapy delivery, different kinds of small molecules, new kinds of antibodies, PROTACs, people who have those ideas and they understand those technologies. And we explain Charcot-Marie-Tooth to them and try to entice them essentially into looking at Charcot-Marie-Tooth as an indication for their technology. So there's some sharing of information there. And when we find a good fit, what we do is we then provide the research funding, but we also provide advice. And so, we get a proposal from the applicant, and then our scientific advisory board provides a feedback on, hey, we love it, but maybe change this piece, maybe make it a 3-week treatment period instead of a 2-week, or maybe consider using the C22 mouse model instead of the C3 mouse model, et cetera. But then once we decide to fund, then it's the researchers or the applicants who then go do the research. But here's why I call it hybrid. I call it hybrid, because of that initial, sort of involvement in designing the study, although we largely give a free hand to the applicant, we're providing advice. But then also we provide the funding in a stepwise fashion. We want to see specific aims that make sense and follow one from the other. So sort of a stepwise research plan. And then what we do is there's always a gate at the end of each step. And we make a go, no-go decision with the funded researcher. And so we get together and they say, okay, here are the results from stage 1. This is largely what we thought it was going to be. It provides great support to go into stage 2. Then we just go into stage 2. But sometimes, of course, what happens is you get a report that says, well, we got a lot of surprises in stage 1. So what we thought was going to make sense for stage 2 maybe doesn't really make sense anymore. So why don't we all put our heads together and come up with a new stage 2? So from that standpoint, we do stay engaged in the project. And that's why I'm just very grateful to have a scientific advisory board that I think is just top notch and world class, and very eager to get involved in helping make some of those decisions, provide that advice.
I guess the most recent decision was the funding for Samsara. Could you give me a bit of background on that?
So this is something we're very excited about. I mean, I guess, of course, we wouldn't have funded it if we weren't. But Samsara is a young company out of the United Kingdom. I believe they also have a base of operations in the US. So they're really a UK/US based biotech company. And what they're doing is they're developing small molecules to enhance autophagy. And that's important because Charcot-Marie-Tooth is a neurodegenerative disease. And like a lot of neurodegenerative diseases, we see that there's protein aggregation and inclusion of these aggregated proteins inside the cells. So if you're familiar with Alzheimer's disease, for example, you have plaques and tangles, both of which are aggregated proteins. It's amyloid plaques and tau tangles. If you're familiar with Huntington's disease, you get inclusion bodies in the cells there made up of aggregated Huntington protein. If you're familiar with Parkinson's disease, you get inclusions of aggregated alpha-synuclein called Lewy bodies. And it looks like something similar is happening in Charcot-Marie-Tooth, where you can get aggregates of some of the proteins that are mutated. And so autophagy, which is sort of the body's natural recycling system to take out the trash, if you will, to recycle some of those damaged or malfunctioning proteins, the theory is that that process normally occurs all the time with these proteins, but then in Charcot-Marie-Tooth disease, that system gets overwhelmed. Well, what Samsara is doing is they're developing molecules that will sort of supercharge that autophagy process. The idea being there that if you can keep up the production of these proteins and stop them from aggregating, then you might be able to reduce the symptoms of the disease, or again, potentially prevent or reverse even the disease. To our knowledge, this is the first company that's developing these autophagy enhancers for Charcot-Marie-Tooth. And this is exactly why we were founded, to try to find companies and academic researchers who are doing new and interesting research that we think has an application to Charcot-Marie-Tooth, introduce them to Charcot-Marie-Tooth, and then provide the research funding to do that research to prove it out.
You mentioned all of the things that are happening in the field. Obviously, you can't be 100% accurate, but crystal ball, what do you think is sort of reasonable timescales in terms of having some of these treatments available?
Yeah, that's a great question. And of course, we know that, most clinical trials, don't end up working out, unfortunately, right? Most of them don't hit their primary endpoints. So I want to be very careful and say that just because something is in clinical trials now or maybe in clinical trials soon doesn't necessarily mean that that particular therapeutic is going to be the one that hits. There have been many disappointments, and there will probably be more disappointments in the future. That being said, Pharnex is the company that has the treatment for CMT1A right now in phase 3, and Applied Therapeutics is the company that has the phase 3 for SORD deficiency, which is a type of CMT. Both of those, since they're in phase 3, they could hit, right? They could hit their primary endpoints. And if they do, I think it's fairly likely that the FDA would approve one or both of them, and then followed, by the EMA probably shortly thereafter. And so if that's the case, we could have some treatments for Charcot-Marie-Tooth within 2 years. There's a potential that we could see these on the market by 2024, or potentially sometime in 2024. Of course, that assumes that they hit their primary endpoints, right? And there's safety and efficacy, which they may not. For gene therapies, I think that's a little further behind. We have rationally designed gene therapies now that are just preparing to enter their first in human trials next year. And so from that standpoint, if things continue to go sort of in an ideal timeline and we don't run into any major hiccups, one or more of those could be approved by, say, 2029, 2030. So the future is bright, but there's probably not going to be a therapeutic for CMT in your medicine cabinet next year. It's probably not going to be in 2023, but potentially 2024.
And now we're able to go over to the US for our weekly chat with JLL. Only this time, it's a revisit to the first guest we had from the company, and that was Robert Coughlin. Great to have you back on the show.
Hey, Jim. How are you? Thanks so much for having me back. This week, I had the pleasure of attending the MedTech conference, which happened to be in Boston. Now, the MedTech conference is the industry trade association AdvaMed, which is the medical device and health tech companies from all over the world. They had their annual conference right here in Boston. And I am so excited to tell you that it was exciting. Right? We all know that the market's not in great shape right now and that a lot of companies are having difficult times. We're coming out of COVID. And, this is the first time that they've had the MedTech conference since before COVID. So nobody really knew how good it would be. Well, I'm so happy to tell you that the attendance was an all-time high. The amount of companies participating was an all-time high. It was really a true example of folks from government, whether it was local governments and our federal government and foreign governments all coming together to work with industry and academia and talk about what amazing science is happening out there in the area of medical device and med tech. Hey, and we all know I spend a lot of time working with therapeutic companies and biotech companies and pharma companies, but we can't lose sight of the fact that it's really the device industry that really plays a huge role in scientific advancements and health and wellness for people. And to see the new products that were coming out were truly, truly amazing. And I guess the thing that I'm the most excited about is that, you know, people really had a positive attitude about the future. You know, you never know how a dip in the markets is going to affect advancement of science and technologies, and we all worry about that. But, you know, I'm as confident as ever, especially now in seeing so many leaders of industry, leaders of the life science industry talking about, hey, you know, times are tough, but we're going to get through it. Just like 2008 to 2012, we had a real tough time with the stock market, but look what happened. It caused us to change how we behave as companies and get better at it. And I see that again. You know, we talk a lot too about companies cooperating and collaborating and working better as a result of Operation Warp Speed and everything that happened as a result of our world trying to create, you know, vaccines. Well, guess what? The scientists and the companies are all doing the same thing now as it relates to medical devices and technology and replacement parts and surgical instruments. So for me, I just can't tell you how excited I am to come out of this conference this week and see how amazingly successful it was. And I tell you, the future is bright for patients, and that's why we all do what we do.
Okay, great. Thanks for that, Robert. Sounds like it was a great event, and it's always good to be in Boston. Robert Coughlin leads the New England Life Science Practice Group for JLL, a global commercial real estate services company shaping the future of real estate for a better world. He has over 14 years of experience in the representation of lab, GMP manufacturing, and technology space. He is a past president and CEO of MassBio, where he advanced Massachusetts' leadership and advancement of the life sciences industry. And that's it for another week and for another podcast. This time next week, Halloween will have come and gone. Everyone hopefully is home from BioEurope, and I'll be preparing for a trip to Copenhagen. And it will be November, and that will mean I'll have even more videos to edit. The interviews are already done, although I may slot a couple in from the recent events. Once I've got them all sorted out, that is. So I hope you enjoyed this week's podcast, and wherever in the world you are, you have a great week ahead, and that you'll join us next time for another Beyond Biotech.